The first segmentation genes to be expressed in the Drosophila embryo and the first zygotic genes, in fact, are the gap genes. As you can see, the expression of these genes, which are all transcription factors, appears as a pattern of broad overlapping stripes. There are four major gap genes, which include the gene hunchback, which is also a maternal effect gene, as well as the genes crupple, giant, and NURBS. If all four of these genes were deleted, the entire segmented portion of the fly would be missing, since each of these genes are expressed in broad regions of the fly. The expression of the different gap genes is determined by the maternal effect genes. So bicoid and caudal and the hunchback protein that was transcribed from the maternal mRNA are important for initiating the expression of the different gap genes. And later we'll see how the gap genes actually regulate each other as well. As you can see, bicoid, which is located in the anterior region of the embryo, acts as an activator for the genes hunchback, NURBS, and giant. And caudal also acts as an activator of giant. So you see that giant is expressed in two different regions and is shown here in the green along the anterior posterior axis of the embryo. Its expression in the anterior of the embryo is activated by bicoid, while its expression in the posterior is activated by caudal. What do you suppose allows this gene giant to be expressed in these two different regions of the embryo, anteriorly and posteriorly, even though these two regions have very different properties? Recall that enhancers are what determines when and where a particular gene is expressed. So this gene giant can have different enhancers for different regions. So it could have an anterior enhancer and an enhancer that allows it to be expressed in the posterior. The enhancers that are located in the anterior regions would have binding sites for bicoid. So in the anterior region, bicoid and hunchback activate the expression of giant by binding to an anterior enhancer. In the posterior, the enhancer that drives expression to that region would be activated by caudal, which is present in that location. The expression of the gap genes is initiated by the maternal effect genes, but it is maintained by the interaction between the different gap genes because these are transcription factors and because at this point the embryo is still a syncytium. There are still nuclei that are sharing the same cytoplasm. If we look at the regulation among the different gap genes, we can see that the gene hunchback is expressed in a broad region at the anterior of the embryo, which we also saw in the previous lecture. Hunchback and NURBS inhibit each other's expression. So NURBS expression only increases where the levels of maternal and zygotic hunchback begin to drop along the anterior-posterior axis of the embryo. At high concentrations, hunchback also acts as an inhibitor of the gene crupple, but crupple is activated by lower concentrations of hunchback. This is why its expression increases about halfway through the embryo, where the concentrations of hunchback begin to decline. Another interaction that occurs is between the genes crupple and giant, which act as repressors of each other. The interaction between the different gap genes results in a pattern of mRNA expression where there is overlap between the mRNAs of these different genes. As the mRNAs are translated, the proteins diffuse from their site of synthesis, and this contributes to regions of overlap among the different gap proteins. This is shown in the staining where hunchback is labeled in red and cruppled is labeled in green. As you can see, some of the nuclei appear yellow, and that's due to overlap in the expression of these two genes. Another important thing to point out about the expression of these genes is that they contain redundant enhancers. In other words, they contain multiple enhancers that drive expression to the same region. So there are multiple enhancers that drive the expression of crupple to this region of the embryo. And the same thing can be said of the other gap genes. That helps to ensure that the pattern that we see is maintained if there happens to be a mutation in one of the enhancers.